DNA replication is the process of producing two identical replicas from one original DNA molecule. DNA replication is also called a semi-conservative process, which means that each daughter DNA consists of half parental DNA and half of new DNA. We start this process off with the double helix DNA, which is then separated by protein helicase. The two strands are held separate by single-strand DNA binding proteins, or SSBs. Then DNA polymerase III arrives and brings with itself the leading strand. This leading strand pairs with the nucleotides of the original strand and its complementary bases. However, on the lagging strand template, a primer synthesized by primates is added to the nucleotide of the strand brought by DNA polymerase. Because they are not yet together and are fragmented, they are called Okazaki fragments. DNA polymerase I then arrives and switches any RNA nucleotides to DNA nucleotides. Finally, DNA ligase pulls together the Okazaki fragments.
Transcription. Transcription is the synthesis of RNA using information stored in the DNA. Transcription can be divided into three stages: initiation, elongation, and termination of the RNA chaining. It begins with a specific section of the DNA known as the promoter, commonly including a Tata block, a nucleotide sequence that only has thymine and adenine bases and is the starting point of transcription. A collection of proteins called transcription factors attach to the promoter region. After the transcription factors have attached, RNA polymerase II binds to the promoter region. Once completed, the polymerase unwinds the two DNA strands and starts transcribing the template strand at the starting point. As RNA polymerase moves along the DNA, it untwists the double helix. It adds complementary RNA nucleotides to the 3' end of the RNA as it moves along the DNA strand. The growing RNA is trailed by the RNA polymerase and continues until the RNA polymerase reaches the end of the transcription unit. Soon after, proteins cut the RNA strand from the polymerase and releasing a pre-mRNA. Pre-mRNA is sent to RNA editing. The pre-mRNA receives a 5' cap on the 5' end. The 3' end receives poly-A tail. These caps serve to facilitate export of the mature mRNA from the nucleus and help protect the mRNA from degradation. The pre-mRNA consists of exons, genes expressed, and introns, genes unexpressed sections. Through the use of a spliceosome, the introns are cut out and the exons are spliced together. Small RNA within the spliceosome base pair with the specific nucleotides of the intron. The spliceosome cuts the pre-mRNA and splices the exon. The introns are released for degradation and the formation of a messenger RNA is present.
Translation. The whole point of translation is to build a polypeptide by reading a genetic code, and the translator of which is tRNA. The ribosome, made of RNA and proteins, adds each amino acid brought by the tRNA to the growing end of a polypeptide chain. We start with all the components in the cytoplasm. The top and bottom subunits of the ribosome are in place, and the tRNA with codons and the amino acids are present. To the right we have our tRNA, and each anticodon, the bottom, is unique to it. And drawing our attention to the green circle, this is the amino acid attachment site. To the right we have our amino acidyl tRNA synthetases, which provide specificity and covalently joining amino acids to other tRNAs through hydrolysis. This now charged tRNA can deliver its amino acid to a ribosome. At this point, I want to point out what a wobble is. It is defined as a flexible base pairing at a codon position. Assuming at circle 1 we had AGA and at circle 2 we had AGG as the codon, the anticodon carried by the first tRNA, UCU, could pair at either site. Continuing this process, we start building a polypeptide chain with peptide bonds. Now, bringing our attention to the structural components of the ribosome, we have 1. The E site, where discharged tRNAs leave the ribosome, 2. The P site, which holds the growing polypeptide chain, and 3. The A site, which holds tRNA carrying the next amino acid to be carried to the chain. This process will continue until the ribosome reaches a stop codon, which signals the end of the translation process. Gene regulation is maintained by the operons. Altogether, the operator, promoter, and the genes they control constitute an operon. I will be using the lac operon to display the operation of operons. We start with our DNA strand. The RNA polymerase reads the instructions from the gene and creates RNA, which in turn creates protein, the inducer on the left, and enzymes on the right. The mRNA, which reads 5 to 3, creates the inducible enzymes. Looking closer, we see introns and exons. The introns are excised and the exons are spliced together. Allostere, the inducer, binds to the protein or the enzyme. If it binds to the enzyme, both are used up. If it binds to the protein, it creates an inactive receptor. However, the inducer that has not been inactivated binds to the operator, making the RNAs unable to transcribe.